Morning, everybody. Welcome back to the international webinar on plant functional biology. Today, the second day, we have three invited speakers. 10 to 10:45, Professor S. Rajagopal of Hyderabad University, and 10:45 to 11:30, Professor Josh Tiputur of Calicut University. 11:30 to 1, we have paper presentation. Dr. Chandramohan, Head Department of Botany, Brunner College, may chair the session. And we have an evening session by 5 to 5:45 by Dr. Sujit Pudyavitil. And now I invite Dr. Gayatri R. Nambiar to introduce Professor Rajagopal.
Good morning all. Myself Dr. Gayatri R. Nambiar, Assistant Professor, Department of Botany, Syed College, Taliparamba. I am vested with a task to introduce Professor Rajagopal Subramaniam, Department of Plant Science, School of Life Science, University of Hyderabad. Most of the participants in this program are well aware of Professor Rajagopal, who is an eminent personality and scholar in the field of photosynthesis. We are very fortunate to have such an expert in the field with us to spare his precious time and knowledge with us. Yet, for the sake of formality, I would like to give a brief introduction about Professor Rajagopal sir. Professor Rajagopal sir has been working for over 20 years in the field of biophysics and bioenergy and has published 95 research articles in reputed journals. He had 7 years of postdoctoral experience in different countries like Biological Research Center, Hungary, University of Quebec, Canada, and Arizona State University, USA, before he joined at University of Hyderabad. Professor Rajagopal made significant contributions to the area of photosynthesis with emphasis on the bioenergetics and acclimation of chloroplast. His group studied in detail the structural and organizational dynamics of photosystems and their light harvesting complexes in Chlamydomonas reharditi as well as Arabidopsis thaliana. He studied influence on high light and iron deficiency on the organization of photosynthetic apparatus and proposed a structural model of photosystem 1 from Chlamydomonas reharditi. Currently, his lab is engaged with proteomics and metabolomics based approaches to dissect the abiotic stress responsive signaling events in the chloroplast. I cordially invite him for delivering a vibrant speech on a very interesting topic, abiotic stress influence of photosynthesis. Thank you.
Introduction. Hello, can you hear? Can you hear?
Yeah, it's clear. It's clear.
Can I start?
Yeah.
Yeah, go on, sir.
Okay, so yeah, thank you for introducing, and I'm delighted to be here with associate with you, and thank you for the organizers, Dr. Shakira and Abu Salam, who invited me for this webinar series. I'm really happy to associate with you, and also I'm very happy that I'm sharing the platform with Professor Govindji. I consider Professor Govindji is my grandfather, because why I'm telling this one, my guru is Professor Prasanna Mohanty. Yeah, Prasanna Mohanty did PhD with him. I always consider Professor Govindji is my grandpa. You agree, Professor Govindji? Hello. Okay, this is the topic of the today on abiotic stress influence on photosynthesis. Okay. Can I go ahead?
Sir.
Hello. Can I go ahead? Hello.
Yeah, you are audible.
Okay. Okay. The topic of the my part of research is abiotic stress influence on photosynthesis. Yeah, before starting, I would like to thank my funding bodies from Indian organization, DBT, DST, DST FIST, and now the CREB and UGC ISF, that is an Israel project, for financial support. You can see my collaborators from India, Professor Raghavendra, Professor Taliparamba, Reddy, Ramachari, and overseas, Petra from US, Jozo, Peter from Hungary, Ichiro Takahashi, and Nathan Nelson, and Govindji. And we have a paper with Govindji on state transition, which he has discussed yesterday. So these are my students, PhD, past PhD, present PhDs, and in which one of the students, Ms. Pavitra, is from Calicut University, just photo student. And I have a couple of postdocs working in my laboratory. Okay, to start with about origin of photosynthesis, most of you know that, how the photosynthesis evolved, but just to briefly tell about the photosynthesis. So you all know that if you see that left panel, when the about 4.6 billion years ago, we can see the globe is like a hole, this round, the globe shows a kind of a red picture, nothing was there on the earth. Then started with, you can see the left panel, all the volcanoes and other things started, and still there was no oxygen. And the right panel, when you see here, a lot of the water and the ocean formed, and then could able to the plants. You can see the green part, and which we are enjoying with all lush green, the forest and other things, and the globe is the full of oxygen because of the forest, because of the photosynthesis. So whatever the advantage which we have now with the oxygen is all because of the plants. We all know that. So further, the importance of the photosynthesis, we all know that, it is not only for oxygen. You get a lot of, the light panel, all kind of, the dependence of the, the meat, whatever it results from the photosynthesis. Even the lot of the phytochemicals, which is all from the, the derived from the plants, that is through the photosynthesis. And of course, we get all the food, with the right panel, you can see these, whatever the food which you are eating is all because through the photosynthesis reaction. So now recent days, the natural gases are almost going to be over, and through photosynthesis reaction, we can have alternative fuels like hydrogen energy, ethanol, bioethanol, other things. Okay, these are some of the importance. But what is happening these days now, like places like, you can see that it delegate, the pollutions. The pollution, environmental pollution, is influencing photosynthesis. When the climate changes occurs, you can see here what is happening. The strong storm, these are the ones, and it is strong storms, high temperatures, and drought is increases, and snow melts, and all these, the damaged corals in the ocean, and rising sea levels that is happening, and warmer ocean. These are the things, and changes in plant life cycle that happens when the climate changes. That is very threatening these days. Okay, so when it is, one of the factor is a drought. So drought, you see that left panel, about this one, maybe in 2040, in India also, we are going to get a severe water deficiency that is called drought. And our world also, most of these places you can see here, all of them are getting affected, but India is more, will be severely affected if it continues the same kind of pollution and the environmental stress. Okay, to start, the little bit about photosynthesis, we all know that. Professor Govindji, I don't need to talk about photosynthesis, but just to introduce myself and what I'm going to talk about my research. Here, when water, whatever the photosynthesis reactions takes the light, then utilize the water and you get the whatever, through the photosynthesis reactions, electron transport, you get the NADP, ATP and NADPH. And that will be utilized in Kelvin-Benson cycle in order to get the whatever so-called we call the food or starch, glucose, and through that you get all the biological biomolecules like even protein, the amino acid, protein, carbohydrates, and the fats are all will be synthesized through the photosynthesis reactions in the dark cycle. So if you talk about the thylakoid, the membrane architecture, so we have the major membrane complexes, that is the photosystem 1 embedded with the light arresting complexes, that is called LHC2 and photosystem 2. The electrons are transferred to the PQ pool, then transfers to the the photos through PC to the photosystem 1, and finally you get from the FNR you get the NADPH, and through the proton motive force you get the ATP. And also here, sometimes and more when the cyclic electron transport is also will be there around the photosystem 1. So then the structures of photosystem 2 and photosystem 1 has been very well established by these, many groups, but these two structures are from, I'm showing from the from Renshen and Petra from a group. And you can see the left panel photosystem structure has been well organized structure is about 2.5 angstroms. And the photosystem 1 structure from Senecosystis, that is about, the trimer, you can see this trimer here, and that has been well organized. And this is very important, structures are very important to understand the mechanism of photosynthesis. Then also the photosystem 1, the recent review from the Nathan Nelson, and summarized from all the photosystem 1 structure from Senecosystis, as well as the P structures. P structure is from his group, and Rhodosodomonas, bacterial photosynthesis organisms, and Senecococcus, that is also from again Nathan Nelson structure. And the recent structures, the Chlamydomonas has been, PS1 structure is a bit different from the higher plants, which I am going to tell about little bit this ones. When you see the photosystem 1 structures, you have a 2 rings, 2 layers on the, around the core complexes. Core is the one, this one, and over that, the half moon shape, 2 layers will be the light arresting complex are covered. The energy, whatever transfer from through the light arresting complexes from one layer to another one transfer, then it goes to the light arresting. It goes to the photosystem 1. The Chlamydomonas have a different, little bit different here. It is additionally, wherein higher plants, they have only 4 light arresting complexes, whereas in Chlamydomonas, it is only, it has about 10 subunits of light arresting complex. That's why the ratio, if somebody asks me a question, what is the ratio of photosystem 1 and photosystem 2 in Chlamyd? In higher plants is different, but Chlamyd, it is like almost 1:1 ratio, the light arresting and photosystem 1. Okay, then going to the different abiotic stress, how they influence the plant. If you look at with the start from the left panel, salinity is the one, you can see in Kerala also, most of the coastal places, the salinity is a very threatening factor influence the crop yield. So when all the stress, the effect of stress onto the plants may not be same. So each stress have a different characteristic effects. Salinity, if you see the bottom panel, those are, disturbed osmolytes, ion homeostasis, membrane damage that can occur. And heat, generally, again, the common phenomena here, most of them is photosynthesis is reduced. And heat is, the Vinoc and also the heat and drought is almost kind of similar, but, you can have the transpiration will be more in heat. Hence, then water deficiency will occur. And drought also will be photosynthesis less and water stress. And chilling stress is another one. Maybe we don't see in South India much chilling stress, but if you go to North place, Himalaya, those places, you can see decreased rate of biochemical reactions. Other things will happen. Ice crystal mediated damage can also happen. The other one is, which we see in most of the entire across the globe, the light stress, highlight. Highlight, it inhibits photosynthesis, and photooxidation will be the more one you can see, and elevated species generation of different oxidative species. That is very common in highlight stress. And now heavy metal, there will be many protein damage will be there, and the submerged, then you can see many other things, anaerobic reactions. The upper panel, there are also good things. Plants always, they acclimatize, to the different stresses, various stresses. You can see various panel, salt stress, osmoляtes will be more, heat stress, heat shocks will be there to protect the plants. And in drought stress, the stomatal closure will be one of the very important factor, to protect the plants. And chilling stress, we have a different kind of thing. Likewise, there will be acclimatization can occur through balancing from down regulation to up regulation of different proteins and genes. So this is the slides. You can see how the plants are getting affected in different seasons. So you can see with the leaves will be see the fall, then it goes to the green, it affects the highlight, then it goes to a lush green in the after springtime, then flowering time, then likewise various spectrum, then exposed to senescing, then exposed to highlight, different kind of the stress. You can see this is the cycle. But in this cycle, there will be a lot of the factors, the influence. Influence means the biotic and abiotic stress. Okay, this slide shows the entire whatever the spectrum of light cannot be absorbed by the light. Only the limited amount of light can be absorbed. That is the low intensity of photosynthetic active radiation. The rate when it get the bottom of the panel, it gets you can see here up to here it will be active. After that this is the excess light which can be damage to the photosystems. Okay, these kind of stress mechanisms, there is always stress is two types. One is a short-term duration and long-term duration. During this time, they can adapt or acclimatize the plants through all these mechanisms. You can look at this one. The commonality of
No, he's speaking. It's a mute, unmute, I don't know.
Dr. Rajagopal, you are not audible now.
all this one is
Yeah, you have to give the slide share once again. The slide share has gone off. You are in a muted. Yeah. Now you are in a muted state. Please unmute.
hello, can you hear?
Yeah, now it's fine. Now it's fine.
Yeah. Okay, it happens like that. I don't know. There will be a time limit maybe. Okay, so the short-term duration and long-term duration, you can see that the long-term duration, there are several things can happen. These are the ones, protein degradation, change in antenna size, change in PS2, PS1 stoichiometry. And through that one, accumulation of antioxidants can happen, increased. These are the down panel is the non-photochemical punching. We will discuss that one more in a few minutes later. Okay. So now if you see this, all these changes, photosynthesis is very sensitive to change in environment. That is the one, whatever I was talking, and the in specific stress will be, the ROS will be increased, and the light, when the more light is there, you get a photo damage, that through the oxidative damage, and mineral deficiency, you get some of the, some of the important co-factors for photosynthesis, thus photoinhibition happens. And elevated temperature ROS, again, ROS will be more, and chilling stress can result in nutrient deficiency, water deficient, and reduced whatever the electron transport is. I'm going to talk about little bit more on non-photochemical quenching. So that will be connected to my research. Here, the non-photochemical quenching, right panel shows the state transitions, and the left panel shows about the specifically non-photochemical quenching, that is the one delta pH increases, and the down panel is more of the photoinhibition, that you can see the damage, and the upper panel is again the light harvesting complex stress-induced protein, which is recently been reported about almost like 10 years back. Now it is this field is picking up very fast. Through this one also the acidic pH will increase, and that because of acidic pH, the LH2SR3 expression will happen. From left to this up to here, these two are non-photochemical quenching. The right one is the state transitions. The bottom one is the QEI, that is a photoinhibition. All these considered as a non-photochemical quenching. What happens in the plants? What happens in structural wise, if you see this one, if you see this, the trimer of the LHCs, you can see this one, you can see down, the vilazanthin, these are the one, vilazanthin, this can bind to small subunit and the S subunits, M subunits, zeaxanthin, and there is a monomers, that is CP26, 24, 29 will be there, and CP43, 47, they will be there. When it going to the highlight stress, what happens, you can see here in the light panel, when it increases the non-photochemical quenching, there is a protein called in higher plants, PSBS, one of the protein, you can see the blue color here, left panel, that become, when it, in the original state, it will be dimer. When it activates, right, that will become monomer. And that monomer is a dangerous, it is not dangerous, you can say that it's a protective mechanism when it is non-photochemical quenching appears. Similar way, in chlamydomonas, almost similar, but here, apart from the PSBS, there is a LHC SR3, that is acid light harvesting subunit, stress-related proteins. There are three genes. So LHC SR3 is more prone, more always very active when the light is more. That is a highlight conditions. You can see here. That when it gets activated, the delta pH increases, and can see the non-photochemical quenching. Our group focus more on the these are the topics: state transitions in Arabidopsis and chlamydomonas, and acclimatization to the different stress. And during this process, how the thylakoids get organized through PQ pool reduction. And the other area is we are working on protein ligand interactions. So we'll be, for today's talk, I'll be focusing on one is the state transitions and photoinhibition, if time permits, little bit on nutrient deficiency. State transition, most of you know that when it is you can see this preferential excitation of PS2 leads, reduction of PQ pool. Then PQ pool gets reduced, and when it binds to the QB site at the cytochrome B6F complex, and the LHC2 get phosphorylated, that through the LHC2 kinase. And these are the two genes, or two proteins are very important. That is STT7 in chlamydomonas, STN7 is in Arabidopsis. These kinases are activated, and the LHC2 gets phosphorylated, and that will be migrated to photosystem 1 that you can see in next slides. So like this, likewise get phosphorylated and get migrated to photosystem 1. That is a very important process in state transitions. So when it get, again, it cannot be stable. It has to be balanced for photosystem 2 to photosystem 1. The excitation energy has to be balanced. When photosystem 2 oxidized, then it goes to again to photosystem 2, that they get dephosphorylated and migrated to photosystem. This is the cycle, like this. Okay, now what we did here in Arabidopsis, we have taken Arabidopsis here. We grow the plants, and we have given a different lights, a blue light and a red light. One is preferential excitation photosystem 2 and photosystem 1. When we have given a short term, it is like 15 to half an hour is more than enough to induce these state conditions. Of course, we can also give different other the methods also, aerobic and anaerobic conditions also can, that also will show you. So what happens here, when it gets, we have taken an anti-phospho-309 is the antibody to check the LH2 phosphorylation. That is a biomarker for checking the LH2 phosphorylation. So here, when you see here, the B1, B1 is getting phosphorylated, and B3, also getting phosphorylated, and LHC2, B2 also getting phosphorylated. That means the trimer of LHC2 is getting phosphorylated and phosphorylation instead to excitation. So, but we wanted to see why, which one is more active. This was one of the first report from our laboratory. The LHC2 getting phosphorylated, but which one is the first, is getting phosphorylated? That we showed first time. You can see here, this is the state 1. I mean, this is the upper panel is, the state 1, and these are the, state 2 conditions. These denotes the photosystem, light arresting complexes, and these the light arresting complex in state 2. When you see here, 1, 2, 3 major bands, and when you see here, 1, 2, 3, 4 bands appear in 2D electrophoresis. What is this one? The additional band, what is the one? Then we have identified, again probed with the anti-phospho-309. This band belongs to the entire one belongs to the LHC2. LHC2, the B2 subunit. We have a 3 major subunit, 1, 2, 3. The 2 is getting phosphorylated first, and mainly because of 2, that other 2 also getting phosphorylated, and that is when it is, PQ is reduced, then that is moving to the photosystem. That's what we are going to show. Okay, whether they are moving to photosystem 1 or not, to show that one, we have isolated thylakoids and run them to the sucrose density gradient certification. You can see here wild type, we have a mutant that is an LH2 kinase mutant. And these 3 bands, photosystem, this is the light arresting complex, this is the photosystem 2, this is the photosystem 1, LHC1, a super complexes, along with the migrated LHC2. You can see here, and the band is little bit increased, and where in the state, in STN7, you don't expect, because there is no kinase engine. The kinase is absent here, you don't expect any change here. And we have measured the absorption spectrum of this one. I know this is, with the literature, it is matching. When you see this one, density we have, density, we plotted the density of these peaks. State 1 and state 2, we can see the sharp peak, that means it's increased. So this increase is because of what? To want to check, we have checked here, S2, you see this SDHL, the additional band appeared in state 2, that is what we say that LHC1 band. So which is clear that the LHC1 is migrating to, LHC2 is migrating, LHC2 is migrating to photosystem 1. So, and again we probed whether these bands are all over the fractions, or they're sticking to only one place. It is, you can see here in this clearly, it shows in the right side in state 1, it is missing in with the photosystem 1. This is the photosystem 1 band. You see here, no pure LHC2 bands here. When you see the state 2, you can clearly see these LHC2 bands are migrating to photosystem 1, LHC2 1. That means the state 2 PQ pool is reduced, then migrated to photosystem So this also showed with CD circular dichroism spectra. In the mutant, nothing, there is no change here. When you see in the wild type, you have clearly the peaks have increased. That means the LSC2 is migrated here. That means photosystem 1, photosystem 2 to photosystem 1, LSC2 migrated. Hence, you can see the increased signals. Similarly, the state transitions is only stick to the light. No, it is also shows in temperature and in anaerobic. Anyway, anaerobic is there, and also it also shows in the other stress conditions. And again here, I don't have much time to explain you more because the time is limited. Here you can see here the different duration of times, different days, different the days of the plants. We have probed with LSC2 antibodies, and we have also probed with some other D1, D2 antibodies. You can see clear LSC2 is getting phosphorylated, and you can see this low temperature fluorescence. So fluorescence, you can see here the photosystem 1 is increased. That means the LSC2 is increased. But whereas we don't see in the mutant is almost normal. That is it. That means even in the elevated temperature, it mimicking the same like light-induced state transitions. The mechanisms are little different. In chlamydomonas also, we have showed, here also it's bit, it may be little different, but it not going to the 40 degrees Celsius, but here is only up to 38. They could able to induce state transitions. But unfortunately here, the LSC2 is getting phosphorylated, but the LSC2 is not migrating to photosystem 1. Still we are working on these, this topic, but why it is not migrating, because we also know, even I have discussed with Professor Govindji and other people on a broad platform. Now there is a spillover mechanism. Through this spillover mechanism, it is getting balanced from photosystem 2 to photosystem 1. Similarly, we showed with a different, the sucrose gradient. The sucrose gradient shows that the photosystem 1 is, the LSC2 is not migrating to photosystem 1 in light in the temperature induced state transitions. Whereas when it getting relaxed, we relax them again, the peak pole is getting reduced, and again the LSC2 getting phosphorylated, and here LSC2 is migrating. The reason we don't know, because we have not yet gone through these results yet, but these are new results. So what we said here, the model which shows the PQ pool is in temperature, in highlight, in the fluctuating light, or in the light which is in elevated temperature in Arabidopsis and elevated temperature in chlamydomonas. Whatever here is if it is you can see the state 1 to state 2, the LSC2 is getting phosphorylated and migrating. May not be, it is attached, maybe this is through with the spillover mechanisms. But however, when it is in chlamydomonas, when it goes to the state 2 in relaxation stage, that will stick to the state 2 conditions. And similarly, it shows in the anaxigenic conditions or anaerobiasis. In Arabidopsis, we have showed it is also mimicking to the light-induced state transition. This is well-known phenomenon. Okay, the next one, which I am going to discuss about the light-induced highlight-induced changes in the photosystems and through the non-photochemical quenching. We have taken here is a chlamydomonas is a model system, which is easy to grow, and you get a lot of mutants, and we can also make a lot of mutants from this one. I'm just we grow the chlamydomonas. Chlamydomonas is a beautiful system. It can grow in different photoheterotrophic, photoautotrophic, and mixotrophically also they can grow. So for photoautotrophic, we use a TAP, acetate medium. HSM is a regular high salt medium, but they grow in the autotrophic growth. And here, when we used different light conditions, 500 micromoles per meter square, and 1000, and the similar way in highlight also we have used. here, what we observed is a faster growth when your light is increased and you have up to up to 500, they grow very fast. And 1000, initially there will be less, but after 2 days they pick up very fast. They also grow very fast. Okay, but similar way in HSM it behaves differently because their autotrophic growth. From initial growth onwards, they're very faster than the acetate medium. And what is you can see this one, we have put them in the agar plates and also checked whether how they're sensitive. We have a different mutants, those to want to check the difference between linear and cyclic electron transport. These two are left panels, the wild type chlamydomonas, and these two right panels, these PGR1 and PGR5, proton gradient 1 and proton gradient 5. These are the cyclic electron transport mutants. And we put them into different light conditions here, 50 to 500. And you can clearly see that, in 500, the growth is inhibited here. And whereas in the HSM, also similar HSM, it is not even growing in 500. They are almost, damaged. That growth is very, very less. And a similar way you can see here in the bottom. So you see no growth in PGR5. PGR5 is a very sensitive mutant because the cyclic electron transport is very, very, very important. And also the PGR5 also involved indirectly with the non-photochemical quenching. If there is no non-photochemical quenching, they may not, they're very susceptible to light. And we got through some, the oxygen evolution measurements where in the mutants, their oxygen evolution is reduced. However, when you see the protein level, Western blotting, the PSBO and P, they're not changed much, even wild type and PGR1, but a little bit, at a PGR1, there is a small change in the oxygen evolution. That's why you can see that activity also not that much change. But PGR5, I'm not showing here, PGR5 is very sensitive because I said this PGR5 is directly linked to the NPQ because it's NPQ and as well as the cyclic electron transport, both of them will be absent. Hence, there will be more damage to the algae. And I think most of you know, Kanur and other, Jas and other many other people in India, they're measuring the HandiP to see the photosynthetic efficiency. You can see here the curves, OJIP curves, and it's a change in different, and when you increase the light, they change it, and you can see here the Fe bar FM, and even also Fe bar FM mutants. The mutants, the photosynthetic rate is very, very low. The mutants, because I said, because cyclic electron transport is very important. And also we used the light-induced curves, and you can see here the left panel photoheterotrophic and photoautotrophic. We use various light in the PAM measurement. These are the PAM measurements. The electron transport with the different light induction curves. What it shows here, the little bit variation is there electron transport, where you can see here, mutants, their change electron transport is significantly affected. And here also the efficiency of photostorm 2 is reduced in the mutants, but not in the wild type much. And interesting, and all of you are not thinking, most of us, I said, highlight, when you shine the highlight to the plant, but here is the algae was grown in light condition itself from the beginning. It is not exposed and it is not treated in the, after the mid-lock phase. People used to do that during the mid-lock phase. They expose the algae to the highlight, so where people see that. But here is, we have grown these cells from the beginning, where it is clearly, you can see these in wild type, in highlight conditions, the NPQ is increased. That is just what we always expect. And similarly, it is here, and HSM is not much, but there is a small increase. And the other one is, if you see that, there are two components, non-photochemical quenching, that is, NPQ, YNPQ. The other one is, non-regulated energy loss, that is also you can see in NO, that is reduced instead of increased, that is reduced, very common phenomena in high plants. They are all reduced in mutants much. And why this all happening? So here the chlorophyll AB we have measured, and there is a small change in one, but in a mutant is bit changed. But the other pigments which is causing the non-photochemical quenching, the violozanthin, neoxanthin, lutein, beta carotene, all of them, the beta carotene is increased in mutants, and ultimately all of them converting to zeaxanthin. When you zeaxanthin, you can see here all these one when it light, when it takes excitation, light is increased. You can all these pigments they convert. You can see here right panel, violozanthin, anthrazanthin, all of them they convert into zeaxanthin. Zeaxanthin is the one it quenches the energy. That then increase the non-photochemical quenching. This is the main reason the non-photochemical quenching is increased. And further, what we did, circular dichroism is the one, it tells about the macroaggregation of the thylakoids, and that has been very, very sensitive. That means the protein to pigment, and that's what I can see the pigments has been changed. The protein to pigments has changed. That's why you can see the macroaggregation is completely lost. That's also, maybe just knowing little bit more about this, but the pi type and psi type bands are tells about the macroaggregation, which has been reduced quite very, very enormous decrease here because of the macroaggregation, because protein pigments the ratio has been changed. Then the next one to agreement with the CD spectroscopy with other parameters. What we did, the blue native gels, which have been standardized in our laboratory, that tells about the super complexes. Super complex, which means photostone 1 LHC1s, or photostone 2, photostone 1 and 2 monomers, LH2 assembly, LH2 trimers, and LH2 monomers. We can separate them using detergents, different detergents. Where you can see the right panels, when you give light is more, these higher super complexes have been totally, changed. That means disturbed. And the other, they're stable. The other complexes are stable. That's why, still the plants can, they are, algae can still able to grow. Similarly, we use the mutants. In the mutants is much more damaged. The super complex is damaged much more. That is why you can also see the CD spectroscopy. Because of the super complex change, that means the pigment protein has changed. You can see the CD signals has been almost lost in highlight conditions. We did a lot of protein analysis with using different antibodies. The D1, you can see the stars. They are the one little bit down regulated. D1, CP47, the little bit PSBO in highlight. That means 1000 micron instance mostly. In 500 is not much. And you can also see the trimers, which I showed now, why it is the super, again, connecting to the super complexes of photostone 1 and photostone 2, why they have been disturbed because of the trimer of, the LHC2 is getting down regulated, the star marks. That's why, you are going to see the change in the super complex. Also, there are monomers also changing in highlight conditions. And interestingly, as I told you that, the LHCSR3, that is a light harvesting stress-related gene, is always increased when the NPQ is. That means that it is also connected to the PAM measurements. When the NPQ, non-photochemical quenching fluorescence, is increased, because it's also this protein, LHCSR3 protein or gene, that is one major thing, which is increasing in the NPQ. Similarly, we have probed with all, protostone 1 antibodies. Here you can see the reaction center proteins. Here equally, reaction centers and, even some of the light arresting complexes, you can see clear the stars. Their LHCs are little stable, but somehow the core proteins are more, they're unstable because of the PS1 is the one acceptor side of photostone 1 is a major place where the reactive oxygen species can generate. Those reactive oxygen species can attack the photostone 1 reaction center. They're the first ones to exposed to these reactive oxygen species. That's why you could see more of damage of reactions at photostom 1. Further, what we did, we went for transcriptome analysis, whole transcriptome analysis, that is the next generation sequence based ones, where we can see here, from low light means regular light, and highlight means 500, highlight 2 is 1000 microns. Here, as I said, the growth is increased, but photosynthesis is reduced. But something is there, a mystery. It is still green. It is still green in even in 1000 microns. Some of them, most of the cells are green of the four days of the cultures. So there is something hidden. Things are balancing the cells. The balancing to the or acclimatizing to the highlight. What are those genes? We wanted to understand. Here you can see this one. All these genes. I mean, these are the different folds of the one. The upper panel is upregulation. The down panel is the down regulation of the genes. So in those genes, you can see clearly 2 folds, and you can see that almost a similar kind of expression of the genes in one. I mean, highlight 2, highlight 2. Where in the right panel you can see these. You can nuclear and chloroplast and mitochondrial compartmentalization. So 14,836 genes are expressed. That means down regulation, up regulation together. And chloroplast only 74, and mitochondria are only 8 genes are the expressed. To extrapolate an upregulation and downregulation genes, you can clearly see here these almost. It is say that here from low light to highlight 1 and low light to highlight 2. Here same way low light to highlight and low light to highlight 2. The genes are the upregulation genes and downregulation genes are almost same. Like for example here 615 genes and here is 568. But they're little bit. And you can see maybe the downregulation genes are less, but upregulation genes are 1557 is upregulation, and downregulation is only 732. That is very, very important. And what are all those genes? Just to show you. These are the KSHEC pathways analysis we did. And interestingly, again, compartmentalization, what we did, mitochondrial, chloroplast genes and the nuclear genes. And here, these are the genes are upregulated, and these are the genes upregulated in low light to highlight. But wherever, apart in all these genes, the nuclear genes are more upregulated, which can be explained many things because these, metabolically, they're very active. That's why, the different metabolic genes are very active. That's why it is good able to acclimatize to the highlight. If you can see the down regulation, similar way, the same kind of set of genes, the mitochondrial genes, as well as the chloroplast and nuclear genes are more upregulated. But here also these are all the metabolic, oxidative stress, different kind of then photosynthetic genes, and by some of the nuclear genes which is encoded to the mitochondria and chloroplast also it are down-regulated. But what I mean to say that both upregulated and down-regulated genes are almost similar. Then we wanted to extrapolate the only chloroplast genes. So if you say the heat map, the green shows the more of the down-regulation, the black shows overexpression, the red is medium kind of expressions. Where I clearly said that when the stress plants are the algal system, specifically algal system, when it undergoes to stress, you see that here the LSCSR3 genes are overexpressed. So which is also with the protein and also with the genes also synchronizing each other. Then to show that the graphical representation, you can see this. The green is the one with different genes. Now you see that some of the genes, photosystem 2 genes are little bit overexpressed, and you can see these all the light arresting complex genes are down-regulated, which is also again for agreement with the protein data. And similarly, if you can see the photosystem 1 genes some of the genes are overexpressed and some of the genes are, most of the light arresting complex genes are slightly reduced or down regulated. This is all because, again, whatever this one, it is a balancing between photosystem 1 to photosystem 2 because of the genes, which is nuclear genes is playing major role. To summarize this one, so you can see this biomass is increased in wild type when we grow the cells to the highlight, and which is a very important commercial importance. We are looking to this one more to some other, we are extrapolating to different the feedstocks like this. You can use this one feeds because within 2 days the clammy is growing very fast, and the growth is more, and you get more of algae. The algae can be used for several things. For example, you all will be knowing that it can also increase a lot of other metabolites, and also we are looking at it. And also the lipids are enormously increased. That means the triglycerides, specifically triglycerides are increased. That triglycerides can be used as a biodiesel. And most of the light, you can see that LSC genes are downregulated. And now some of the genes are upregulated. That is one, you can see the specific gene relaxin center proteins are, upregulated, and which is indicating a faster turnover to the highlight conditions. And some of the genes, we can see that I already told it many times, the LSC3 genes are overexpressed. That's why this upregulation gene, because of upregulation, you can also see the NPQ is more. Through NPQ, we say that through NPQ mechanism, the algae is getting acclimatized to the highlight conditions. So the last point, you can say the transcriptome profile and their involvement in metabolic pathways provide robust data for further studies to understand. We are still understanding many things with this data. Okay, I think it is already, I think 5-6 minutes or more. Maybe I will, can I stop here or can I continue for a few more minutes?
Sir, we have one more talk after this one.
Okay.
And then this presentation also there.
I'll take the questions. Thank you very much. If you have any questions, I'll be happy to answer.
So now the session is open for the discussion. If you have any question, you can.
I have one, Dr. Rajagopal.
Yeah.
You just mentioned about highlight in biomass enhancement, which may be useful for the feedstock of bioethanol. But is that highlight is in a specific range or very much precise, like 800 moles, or is in a range, or whether?
No, it's a very good question. I would say that the highlight, if you give 1000 microinstance, that is more little bit susceptible to the cells. However, if you use 250 and 500, I would say that 500 is a very optimal. Within 2 days, you get the growth is faster within 2 days. So the 500 is the right one to use for feedstock. And also it's approved. We also use this one. I'm giving this one to the these cultures to the animal biology, animal science, zoology. One of my colleagues is working on fish biology. They're using it, and the weight is about 20% increase in the weight with the control.
So one question from the Siddharth. Does non-photochemical quenching take place in PS system 1? The chat box is there. Does non-photochemical quenching takes place in PS system 1?
You're talking about photosystem 1? No, photosystem 1, you don't see much non-photochemical quenching. Photosystem 2. Photosystem 2 get non-photochemical quenching because there are many reasons. It is when it is a luminal pH is increased, that means it becomes more of acidic. And during that time, whatever I told you, the pigments from lutein, violoxanthin, they're all converted into zeaxanthin, which increases the delta pH. In turn, you get the non-photochemical quenching, and also the expression of light arresting complex stress-related protein also increases. That is called non-photochemical quenching. Is it clear? Siddharth.
Maybe he may be muted, so he will not be able to respond.
Yeah,
Oh.
sir. Next question is from Miss Krishna. And
Yeah,
what is
hello,
the
Krishna.
major role of PS System 2?
Hello. Can you hear me? Oh, you know, it's like in a heart. So whatever with the human beings and an animal system is a heart, the PS2 is a heart of a plant. That is specifically, the it's called, fast recovering protein or fast turnover protein D1, D2 plays a major role to harvest the light energy. The PS1 is, also, both are important, but PS2 is more important because that is the first place to harvest the energy by, from the, solar system.
Okay, thank you, sir.
There are some people
Participants.
with the raised hands in the participant list. I don't know whether they are for a question or I don't know.
Actually, they're raised as in the starting itself, sir.
Yeah,
Okay.
right from the beginning of the talk.
Hello.
Yeah, one more question from the Siddharth. Does NPQ have any damaging effect on plant?
Oh, sorry.
Does NPQ have any damaging effect on plant?
Oh, yeah. No, yeah, I would. No, that's what I said. No, the NPQ is, initially, NPQ, yeah, there is a damage. Certainly there is a damage, but it's also adapting. When, I can, Professor Govind knows very well, but, he's also, when the delta pH is increased, there is a heat dissipation. So when sometimes what happens when NPQ is increased much, and when it goes down a little bit, and you can see the damage to the photosystem. It's also see the damage to photosystem. It can be, even if there is a damage, but D1, D2 will be there, so they can be, they protect the plants. Is it clear for you, Siddharth? Or you can email, I'll be available. I can answer you much better.
Okay, thank you, sir. One more question from Shivkumar Patatil. What was the MALDI-TOF method specifically used for protein identification you mentioned? Did you need to employ some purification steps?
Okay. MALDI-TOF, I didn't tell, but we did a lot of proteome analysis. But this is a methodology what she or he, sorry. Whatever, the proteins has to be pure. You cannot use a raw protein or crude protein. Protein has to be 100% pure for MALDI-TOF. We use protein, we extract the protein, whichever protein can be a mixture of protein, but if it is a single protein, MALDI-TOF is a very beautiful technique because with the data bank, you can extrapolate the data. But protein, when you are giving protein to the mass spec, the protein should be 100% pure. We use a different purification system to purify the protein. Then also we use a kind of a jip tip where in the end, we purify the protein and subject it to the MALDI-TOF. So you cannot use crude protein to the mass spec. And even with the salts and other things have to be removed. Should be completely pure protein. You cannot mix any other biomolecules also. You can see my publications. You can just type, Rajagopal Subramanian. There are many publications on proteomics. Maybe you can watch.
So one more question from the Nobel is how this highlight is applied, whether by applying high intensity light for short time interval or by increasing the time.
No, I didn't get the question.
How this highlight is applied, whether by applying high intensity light for short time or by increasing the time.
No, actually what we did is we have given a light, continuous light. Continuous light is not a short or long light. With a different light intensity, with a different, we use a different flask to grow I mean, we have a different flask or different systems, shaking systems. But one batch will be 500 microinstants, another batch is 1,000 microinstants. Continuously, we put them to the light conditions. Of course, you can also give short light, the short, the fluctuating, it's called fluctuating light. The automatic machines are there, you give a fluctuating light, also can be given. So that is a different kind of strategy. Working on fluctuating light is a totally different subject. Is it clear?
Okay, thank you, sir. Shall we move on to the next session? And I think we don't have more questions.
Yeah, thank you very much. Thank you all of you.
Can I say one thing? Can you hear me?
Yeah, sure, sir.
Yeah.
Well, I want to say that Rajagopal's talk was really exciting and wonderful, and I wish he had given my talk because he did such a wonderful job in his.
Oh, thank you very much.
Thank you, sir. On behalf of Syed College, I extend vote of thanks to Dr. Rajagopal, sir. And thank you very much, sir, for your informative talk. And I